Good afternoon, everyone. Thank you very much to attend to our first session of the From Grapes to Wines Cool Climate webinars. I would like to introduce myself. My name is Francisco Diez. I'm the viticulture specialist at Perennia. And today I will be hosting this session, and co-hosting this session is my colleague, Neslihan Ibit. She is the winemaking and innovation specialist at Perennia as well. At this opportunity, we'll be talking about ripening grapes in Eastern Canada, how climate affects grape potential for winemaking. And this will be presented by Dr. Karine Pedneault. Before starting, I would like to thank Perennia for all their support for these sessions, as well the marketing department that without them, we wouldn't be able to be doing these sessions. Before starting, I would like to do some housekeeping so we are all together on the same page. All participants will have their audio and video turned off during this webinar, okay? If you would like to ask a question, please use the Q&A feature that you will see at the bottom of your screen. The chat and the Q&A will be monitored through the session by Nestle and addressed during the live Q&A session happening at the end of the presentation. So please, I encourage you to be writing your questions. They will be addressed at the end. Before starting, I would like to provide some information about Perennia and our vision, our mission. Our vision is Nova Scotia is a recognized world leader in producing innovative, environmental, responsible, safe food of impeccable quality. Our mission is supporting growth, transformation, and economic development in Nova Scotia's agriculture, seafood, and food and beverage sectors. For the people who doesn't know, Perennia covers different services areas, such as agriculture, providing support to the growers in different aspects as pest and disease with the pathologies or a health plant lab. But also we have other departments such as quality and food safety, product development, lab services, and the last addition to the company, fisheries. Related to grape and wine, Perennia has a specific section, and we can different services to the growers and the producers. And it's called From Vine to Wine. Here, as you can see, we can help you with site assessments, viticulture, plant health lab, winemaking services, mobile bottling and filtration, and development services to create a new product. So we cover from vine to wine. At this opportunity, in our first session, we have different attendees, participants from all around the world. That's why we'll be giving some information about Canada and specifically Nova Scotia. As you can see in this map, we have the country, whole Canada, and we have different wine regions. And we'll mention the four main ones from west to east. So as you can see here at the west, we have British Columbia, and we can highlight Vancouver, Fraser Valley, and Okanagan. Going to the east, we can see Ontario, where we have Lake Erie, Pelee Island, Niagara, and Prince Edward County. Moving slightly more to the east, we have Quebec, and where I highlight Montérégie. And finally, we have Nova Scotia at the end, highlighting the Annapolis Valley. In Nova Scotia, the vineyards have been increasing steadily in the last 20 years. Currently, we have more than 1,200 acres, which is 485 hectares. From all this acreage, and 90% approximately of the surface correspond for wine, and the other 2% for table grapes. We have distributed through the province more than 20 wineries. Some main characteristics of Nova Scotia, it's a peninsula, it's surrounded by water, and I would like to highlight the Bay of Fundy. The Bay of Fundy has the highest tides in the world, and this movement of water produces a lot of air movement, which allow us to avoid extremely low temperatures in winter and permits the plants to survive. And in summer, we don't have extreme hot temperatures neither. Climatically speaking, we have approximately 1,100 growing degree days and around 160 days of frost-free days. And in terms of varieties, it divided in interspecific hybrids and Vitis vinifera. In the interspecific hybrids and in the white varieties, we have L'Acadie Blanc. L'Acadie Blanc, I would like to highlight, it's a variety bred in Vineland, Ontario, but adapted very well to the local conditions in Nova Scotia. New York Muscat, Sylval Blanc, and Vidal Blanc, just to mention a couple of examples. Meanwhile, in the red varieties, we have Maréchal Foch, Marquette, Léon Milot, Baco Noir, and Lucie Kuhlmann. Also, we have more than a couple of Vitis vinifera varieties. In the whites, we have Chardonnay, Riesling, Sauvignon Blanc, and Ortega. While in the red ones, we have Pinot Noir, Cabernet Franc, Pinot Meunier, or just Meunier, and Gamay Noir, just to mention again some couple of examples of which kind of vinifera varieties we have in the province. So before starting our presentation, I would like to thank Dr. Karine Pedneault to join us and to have her here. And I will read a little bit about her city. She is a Bachelor of Science in Biochemistry, a Master of Science and PhD in Plant Biology from Université Laval. She started her career in wine science at a Gallo winery in Modesto, California. And after, came back to Canada, where she developed a huge expertise on the maturity of hybrid grapes varieties in Quebec. She has been author of more than 100 publications. And currently she's leading a project on berry ripening in Eastern Canada within the Canadian Agriculture and Agri-Food Canada cluster on viticulture and enology. Also, she's leading the construction of the first research winery at Université Sainte-Anne, where she's professor. If you want to read more about the CV of Dr. Karine Pedneault and get more information, I invite you to see the information in the website of Perennia. Karine, the screen is all yours.
Great. Thank you so much for such a kind presentation, and thank you for inviting me. This is a real pleasure to be a speaker for you today. So I'm going to share my screen so you can see my slides. Am I allowed to do that, Francisco?
Yes, you're allowed. You can do it.
Okay, great. Seems to work. All right. So yeah, just a minute. We'll put that in full screen. Is it good like that?
Yes, it looks perfect.
Okay, perfect. I'm just going to show the conversation if there are questions and everything. All right, so thank you so much, everybody, for joining us today. I'm going to talk a little bit about the theory of berry ripening, prior presenting some of the results we had in one of the experiments that we've been carrying for, well, last year and this year, actually. And yeah, I have quite a few slides, so I hope you'll find it interesting. So what is berry maturity? We hear that a lot when we speak about winemaking, viticulture. This is really something that has been studied a lot in other countries as well as in Canada. In cold climate, this is a very important question. So grape maturity is not what we would call the precise and accurate physiological stage. So in the sense that we have phenological stages in grapevine, as you probably know, but it's not, there's no stage that is really full ripeness or berry maturity in the sense of that we can hear in different aspects that I'm going to tell you now. So in plant biology, we see berry maturity as a process, a biological process. So for instance, seeds will mature as soon as veraison happens, because the whole goal of this for the plant is to produce seeds, when we look at it from the plant biology side. On the other side, skin and pulp will continue to ripen until senescence, and veraison itself, as it starts, is pretty much like a long senescence of the fruit to make it more appealing for birds, for example. All viticulturists knows that birds are, well, really like grapes, and many of them actually will say that you should harvest when the birds are coming in, because they are pretty good in this, that the fruits are ripened. So in analogy, we will consider berry maturity from the pulp side. So we will consider the pulp maturity, that is, well, an optimal sugar to acid ratio. And we will also consider in analogy skin maturity. So that corresponds to the stage where you have a proper composition in phenolic compound and aroma compounds as well, especially in white varieties like Adi, which I'm going to talk about today. So we seek for aroma and sugar and acid ratio in enology. So different perspectives here. And even in enology itself, berry maturity will determine the wine style, or the contrary. So wine style will determine berry maturity that we're going to pick as a winemaker, for example. So for just a few examples of this is dry whites, for example. We're going to look for a high fruit, high and flower aroma, and still a bit of acidity, so we can have a little freshness to the wine. And in reds, we'll look for a higher sugar and lower acidity, while looking also at extractable tannins for those, well, depending of the wine style again. But you can see that there's a whole range of berry maturity that are still that are acceptable, actually, from the standpoint of winemaking. So so today I'm not going to give you guidelines about what you should be looking for regarding Adi. I'm going to show you how things may vary in cool climate, or metabolites may vary, and the rest is for you to use as winemakers and viticulturists. Okay, so we have different types of maturity that we will consider usually. There's the textural maturity, if the berries are becoming a little bit soft. This terms varies a lot among hybrids. Many hybrids have thick skin, very pectinous pulp. So those are not necessarily softening as much as what you could see in vinifera, for example. Also, there's a technological maturity, which is Brix, pH, TA, so the usual terms. I bring your attention here to the Brix itself, because we use Brix a lot within the industry, as well, most people in the world, actually. So Brix is a measure of sugar, but it also has other components to it. So for instance, organic acid may contribute to Brix itself. So Brix is based on the refractive index of the juice. So those organic acids may affect also the refractive index of the juice. So it may rise a little bit the Brix that you're going to see with your juice, for example. Aromatic maturity. So as I mentioned, it's mostly about skin and pulp. A lot of the aroma compounds will be found in the skin, actually. And the last one, phenolic maturity, which I'm not going to talk about much today, because it's mostly about red wines, but it's still a component that is important for white wine, though. But it mostly concerns seeds and skin. So, and those are not necessarily coming up at the same time and perfectly timed together. So it's always complex. Actually, all those parameters rarely be at the perfect point for, well, at the same time, actually. So there's always a matter of compromises when we seek a perfect grape for a given wine style. So it's never like a full aroma, full bricks. And in the north, we're confronted to the fact that during fall, frost is coming. Oh, winter is coming, as other may say. So yeah, winter is really coming in the fall. So when that happens, well, we need to take decision in the field about harvesting or not, or leave it or not on the grapevine a little bit longer or not. So it's always something, a matter of compromises. So let's talk about a little bit about the grapevine phenology itself. So the grapevine will start its growth during May, around May in eastern Canada, and the development of berries itself will start around June, just after flowering, and it's divided in two parts. So the first part is about berry formation. So we're not ripening yet at this point. So the berries are just increasing in size. So there's an extensive cell division at this point. The seeds are growing, and cells start to elongate as well later on to make the berries bigger. And once we reach veraison, in between, there's a little bit of a lag phase that is going to like a transition moment between those two phases. And at veraison, we enter the berry ripening process per se. So the berry ripening concerns really the sugar accumulation, secondary metabolites accumulation. That includes tannins, anthocyanins for red grapes particularly, and aroma compounds a lot that will all accumulate during that time. Another view of this process here in the first part, just something to remember. When the grapes are green, they're still doing photosynthesis. Okay, so they still have those chloroplasts, so they can actually do the photosynthesis and produce sugar, produce other compounds similar to leaves, actually. So this is during that time that tannins and hydroxycinnamates will accumulate in the berries, and then they will mostly going down until harvest. During the veraison, we'll see what we call the engustment of berry. This is a new term, well, maybe 10 years old, that a scientist developed just to characterize the fact that berries are becoming better to eat, and to winemaking during this time. So this is mostly about flavor compounds accumulation. Sugars will accumulate during that time as well. So, and at this point, the berries are becoming softer, and they look more like what we will expect at the end of the process. So another thing, this is going on in the plant. It's not only about the berries. So there are processes in the plant where the leaves will produce the photosynthetates. They may produce other compounds that will be transported in the grapes themselves. But as I mentioned, during the first part of this process, the leaves and the berries are doing photosynthesis. So they all need that light to produce compounds, actually, and get and grow, actually. So this is only at veraison that the berries are becoming a huge sink. So it means that they will pull the resources from grapevine, and this is a process that is how to say, it's not unidirectional, but it's a very complex process, because leaves will may actually send photosynthetate toward the apex of the shoot, toward the fruit, toward the roots. So it's a very complex process about how things are going, and this direction of the phloem can be also worked out by pruning. So it's just a little little thoughts here that I'm giving you to food for thoughts. Okay, that as you say in English. So okay. So what are the challenges that we have about ripening berries in Eastern Canada? In Eastern Canada, I'm pretty much including Ontario, Quebec, and the Atlantic here. So we're all, oh yeah, by the way, I just want to mention that I borrowed this very nice picture from our host, Nestle. Thank you, Nestle, for this nice picture of our vineyard during the winter. So what are the challenges that we have in Eastern Canada about berry grow itself, and ripening, of course. So the growing season is short. We have frost, not only fall frost, but also winter frost. So spring, winter frost. Yeah, that's a good one. Okay, so spring frost. So we have spring frost as well as fall frost. So all the growing season is included between those two limits. So it's a very important thing to be able to get the grapes ripened within that time, and that involves, of course, grapevine to grow, the canopy to be shaped and well developed. So all these processes, the fruit production process itself, but also the ripening process may actually happen and give proper fruit for winemaking. So there's also a whole lot of challenges concerning winter survival. So the buds, the plant itself, so in some conditions, the plants may die because the climate is temperature is going too low during the winter. So this is a completely different challenge I won't address today. There are people that are much knowledgeable than me studying that question. Also, another challenge that we have is the weather itself during the growing season. So there's a lot, at least here in Atlantic Canada, there's a lot of rain. There are storms. So storms, I never never experienced storms that much until I lived in Atlantic Canada. So I remember a few hurricanes that we got, and so the wind itself is also a stress for grapevine and the vineyards, and it may actually damage the grapevine as well. So let's talk about the growing season. So we have different scenario in Eastern Canada. We have, for example, long seasons in Atlantic Canada, in Nova Scotia particularly. So where we will have a long time without frost, a number of days without frost that is quite high, but the temperature itself is not that high. In terms of the summer itself is not that high. In contrast, in Quebec, for example, we may have a shorter season. So the grapevine will start a little bit later its development process, and might actually be harvested a little bit earlier than Nova Scotia. But the summer is way warmer than what we have in Nova Scotia. So, and still will be accumulating the same number of growing degree days, more or less. So that's an interesting question to see how that affects the grapes. In contrast, in Ontario and south of Quebec, we see quite a long season, a long growing season, but also a higher accumulation of growing degree days, because we're going toward a more continental climate here, and it's a little bit warmer than what we have here when we're living basically surrounded by the sea. Typically, just to give you a comparison of what we see in typical wine area for vinifera particularly. So you may see bud break in March, as early as in March, for example, in France or Spain and or other countries, traditional wine countries, and the vegetative growth can go up to November, and then within that vegetative growth there will be the reproductive cycle that will be accomplished as well. So you can easily imagine that this is a very long process. There's a lot of days, calendar days actually, that are allowed, that are allocated to that process. Here in Eastern Canada, it's a little bit cut, you know. So it means that the plants have to be quite quick, actually. So they will start in May to break their buds and start the leaf grow in May. Vegetative grow will start. Flowering happens in June, and then veraison in Nova Scotia, we see veraison at the beginning of September. It's quite late, actually. So when you think about that time in September, what happens is that the light is going down as well. So while the grapes are ripening, the light here is going down, and well, it might affect photosynthesis, you see. So these are all just things that I wanted to bring to your attention about berry ripening. So here are few of the questions that we've been looking at in my research with my group. So we're interested in to understanding what matters most. GDD accumulation in terms of how much temperature are warm, you know, our growing season length. So how that relationship between the length of the season and the growing degree days accumulation will affect berry ripening. Also, how is climate affecting grapevine metabolism during fruit development? So, are there impacts of this of temperature, for example? I'm so good. I'm doing a question in English. Terrible. How are temperature spikes affecting grape composition at the end? So if you have a warm spring, for example, and a very cool fall, what happens? So these are a few questions we're exploring with my students. So I'm going to present you the now famous greenhouse experiment. So this is an experiment that we designed to study the impact of climate on the biochemistry of berry ripening in Nova Scotia. So this is an experiment that we've been doing in a commercial vineyard in Nova Scotia. So I'm going to explain to you a little bit of what we did with that experiment. So we actually did this experiment on L'Acadie Blanc in a commercial vineyard located in Wolfville. Those were all VSP trained grapevine, and we controlled the pruning, all the viticulture around it, the yield also. So L'Acadie is an interspecific hybrid variety, as Francisco mentioned a little bit earlier. You can see his lineage here, genetic, you know, a lot of different varieties that were involved, specifically French hybrids, actually, that were in the parentage of L'Acadie Blanc, with the final variety here. So so what we did is actually putting greenhouses on top of the rows in the vineyard. Those were built from polycarbonate panels, and we used polyethylene to cover the bottom here and the sides. We put holes in the top of the panel, for rain, and we use oboes for temperature, light, humidity to monitor what was going on in those greenhouses. So we had an experimental design that involved 4 treatments. So for those treatments were leaving the greenhouse the whole season, so that will rise temperature for sure. And also we aimed at fractioning a little bit things using a pre-veraison and post-veraison treatment. So that means that for certain grapevine, the greenhouse was only put on during the pre-veraison time, so prior veraison. And for other grapevine, the greenhouse was set up only after they turned veraison. So all this to try to fractionate the metabolism and see what was going on with that. And of course, we had a control. So just a little figure here to show you that we've been putting greenhouses on these treatments from June 17 at flowering, actually, up to September 4. And then we changed them on September 4 to October 7 to put the the greenhouses on the post treatment and remove it from the pre-veraison treatment at this time. So we've been sampling with liquid nitrogen and the whole thing at 4 stages, actually. So one stages was prior berry, was during berry formation, while berries were still green and hard. And the other 3 stages were around full maturity, if I can say this, in terms of winemaking or industry production of wine. So we've been measuring also a bunch of parameters, physiological data, photosynthesis, stomatal conductance, Brix pH TA in the last 3 stages, primary metabolites, and free and bound volatile compounds. So I'm not going to show you all those results today because there's a lot, but I tried to build a few pictures actually to give you an idea of what was going on with those berries. And we're still actually in the process of getting those results out, but it's super interesting though. So let's see a little bit what was going on. So was the first question is, was the greenhouse system efficient at actually increasing the temperature? So what we saw is that in the all treatment, so the greenhouse being up all summer, from June, end of June to October, they had a significantly higher amount of growing degree days accumulated. But here, it was not significant between pre, post, and control, which is pretty good, actually, if you think about it, because we will not want to increase a lot in those 2 treatments. So we wanted to see if we could create temperature spikes. So I don't have, unfortunately, the data about the accumulation in each half of the treatment. So later, when we will talk again about that experiment, I will show you that. So let's see what happened to the photosynthesis. So in general, this is only between all and control, the comparison here, but at 2 stages, 36 and 37. So those were separated by maybe 10 or 12 days, something like that, between the measurements. And in general, this is it. We just saw that the photosynthesis, transpiration, and stomatal conductance were just pretty much higher in general in the all treatment compared to control. So there was more photosynthesis in the plants that were under the greenhouses. So this is an important thing because it means that the plants are actually working more. So it's a data that is important, that has some importance for the analysis of the metabolites. Here, a little look at the phenology of the berries at each stages. So here, what we see is that berries were quite similar, but when you take a look at later on, there's a, well, it's pretty similar between post and control, where all and pre-treatments are pretty similar. So you can see here how colored are those grapes. So those were covered by greenhouse during the first part of the summer, as well. Same for these, actually, for the, it's just a different harvest stage. But you can see that the grapes that were actually outside of the greenhouses during most of the summertime, remember that we changed the greenhouse, it's September, just got a lot more color in the berries. We thought it was an interesting thing. So it means that things were going out here regarding phenolic synthesis in those berries. So a little bit of the metrics that growers usually know well, Brix pH and TA. So interestingly, we only saw a higher Brix in the all treatment and all the pre-post, and that is actually super logical when we think about the growing degree days. Brix is usually very well correlated with growing degree days, and in this specific example, we see that there was no significant difference between the pre, post, and control regarding the Brix. pH varied a little bit, was a little bit lower in the pre-veraison treatment, but very little differences here. No difference here in the titratable acidity, no difference between berry weight, and no difference between the berry diameter. And this is quite interesting because you'll see that it was not the case at all times. So this is at harvest here, but things were a little bit different in other stages where we harvest. So let's take a look at the overall picture. Yeah, I know this slide is a little bit overwhelming, maybe. I really like that kind of picture because it shows up a lot of data. But I'm going to explain to you a little bit, if you're not familiar with principal component analysis, how you can actually read this. So what we have on the left here is a graphic of the variables that we measure. So it includes the glucose in the leaves, fructose in the leaf, sucrose in the leaf, and organic acids are here in the berries and here as well. We have berry diameters and weight here, and fructose and glucose in berries here. So, so how you can read that kind of figure is by superposing this graph on top of this one. This one represents actually all the samples that we had, so all the treatments. So the treatments are not identified here. They're identified only with the color, but you can see the sampling stage here. So stage 32, sorry, I said 32 in my graph, but it's 33 actually. And stage 36, 37, and 38. So the goal of this figure here is only to show you that if you imagine that you superpose those two graphs on top of each other, so it means that those samples here have more of these variable here, okay? I don't know if it's clear, but basically at stage 32 or 33, all organic acid were in a higher concentration compared to other grapes. In contrast, same here, the sugars and berry weight diameters were bigger, were higher actually in the very last stages where we harvested. So that means globally that actually the berries ripen. It's funny to say like that, but it means that we had the differences between all the stages that we study actually, and we harvest in terms of primary metabolites and berry size. Next figure, pretty much the same. So at stage 33, what I wanted to show you, so this is the very first harvest we did. We saw a big difference between the post and control. And remember, those were not covered with the greenhouse, and those were covered, the all and the pre-veraison were covered by the greenhouse at the same time. And what we saw actually is that at stage 33, those pre and all berries had a higher rate of sugar, a higher rate of berry weight. Actually, the berries were bigger and were larger. But this difference after just actually disappeared because the other they catched up, you know. So they at the end of the season, they were pretty much similar in terms of these variables here. So let's see. So this is what I was saying. So here, whoops, sorry, I left this, but this is only a typo here. So this is to show you that at the end, when we finally harvest the last berries, they were pretty much similar. All treatments were similar in terms of berry weight, organic acid in the leaf and in the berries, because we will harvest leaves as well during this time to check for the relationship between the source and the sink. So all treatments were similar at the end. So quite boring, huh? Because it means that temperature finally did not affect much or grapes. Well, there's other things though. So let's take a look at the aroma here. So, and this is still ongoing analysis. So my student is working super hard to get those results out. Let's talk first about the bound aroma compounds that we analyze in the all and control treatment. Yeah, I'm going to show you only about the bound aroma. So the bound aroma are those compounds that are going to be released during the winemaking. Some yeast will actually release more of those bound aroma. If you look for a yeast, for example, that has a high beta-glucosidase activity, for example. But those bound aroma are like the potential of the berries that is going to be released during the winemaking. So in the profile that we analyzed, we found in l'Acadie Blanc 58 compounds thus far. Among those compounds, a lot of unknowns that are not identified yet, but we have, for example, well, prominent compounds. Benzyl alcohol, for example, phenyl ethyl alcohol are two of the most important compounds in the aroma profile of l'Acadie Blanc. We also have 8-hydroxylinalool, which is another compound, which is a terpene, actually. A terpene that is not as effective as linalool itself, but it's still effective in terms of aroma. Also, we have a few C13 or isoprenoids that were identified and in good proportion in the blend. So so it's just to give you a little bit of an idea of what's in there in the l'Acadie Blanc. Next, what we saw, and that is quite surprising in a sense, is that among those 58 compounds, there's probably 20 that were mostly more concentrated actually in the control compared to all. So can you imagine? So this guy here was more exposed to conditions, it was less growing degree days, lower growing degree days accumulation, less heat, and it still managed to have a high concentration of many compounds that are of value for winemaking. In contrast, though, the treatment all won the race for benzyl alcohol and phenyl ethyl alcohol here. Those are two still important compounds for wine aroma. Those are fruity and floral compounds, so they can really contribute to the wine aroma itself. So so I think this is very surprising results. This is not what I was expecting, but it means that there are things that are going out during pre-veraison that will actually affect berry, the quality of the berry at the end. So we still don't have the results for pre and post. So we'll see how we were able, if we were actually able or not, to modulate wine aroma based on the temperature treatment that we've been putting on in terms of aroma itself. So just another PCA here to show you the huge difference between the all and the control treatment. And we could explain, and that is that we have 41 plus 14. So a lot of almost, well, what, 55% of the variability is explained just with those variables. So it's quite high actually with the PCA. So most of these variables here were in higher concentration in the control compared to the all treatment that won only for these three there. So, but those trees are still important though, because those are the most prominent compounds in l'Acadie Blanc. So a few final remarks before I pass my time. So what we saw from the greenhouse treatment, we were able to increase the GDD in the all treatment compared to the other treatments. We increase photosynthesis and transpiration in grapevine. So it means that we did affected the metabolism of these grapevine under the greenhouse. We found that there was somewhat of an accelerated berry development in the early stages, but those the vines that were a little bit behind from the post and control treatment did catch up after, and finally at the end everybody was quite equal in terms of berry size and primary metabolite concentration at the very last stages, the very last stage where we harvest. L'Acadie Blanc profile also includes a lot of terpenes, C13 or isoprenoids, and floral and fruity volatile phenols, which makes it quite interesting as a wine for winemaking grape. The bond aroma profile was different, as I just showed you, between the all and control treatments at harvest. And the main difference concerned floral and fruity volatile phenols, the benzyl alcohol and 2-phenylethanol. So meaning that there are things going on probably early in the process, because those phenols actually are coming from well, the degradation of hydroxycinnamates that are produced during the first part of berry development. So it's just things that we're actually analyzing now. So just a few acknowledgements. I thank very much Lockett Vineyards that has been opening its door for us, its field, I should say. So they were amazing, leaving us to put those structures in their field. Marcel, the head of viticulture, there has been a fantastic supporter of our experiment, and really thank you so much, Marcel, if you are there. Also, I thanked the Canadian Grapevine Certification Network, Nova Scotia Agriculture, Nova Scotia Research and Innovation Trust, NSERC, and the Grape Growers Association of Nova Scotia, and of course, Agriculture and Agri-Food Canada, I was going to say in French, that supported that study. Just a few pics to show you what we have new in Sainte-Anne. So my research team here, a few postdocs, master degree students and interns that are working with me right now. And they now have a very new winery to work because we did a lot of wine. We did about 40 wines to study maturity again. So from grape to wine in terms of aroma and other compounds. And our lab that is going very well now, because when I arrived at Sainte-Anne, I was barely having, I did not have a lab at all. So it's really good to see that. So thank you very much, and I will appreciate to get your questions. I'll stop sharing.
Thank you, Karine. We have some questions. Mm-hmm. So the first question has two parts. The first part is, what were you saying about food for thought with respect to pruning?
You mean about the, I will go back to that slide, I think. Yeah, I remember. So this slide here, correct? I think so.
Yes,
yeah.
Exactly.
Yeah. So what I was saying here is that the way that, in which the leaves will because you know the phloem in the plant is all connected toward the stem and the root. So there are a few organs that are acting as sink, and other are acting as sources. So once the grapes pass the stage here, veraison, they are a sink, and the only resource provider is the leaf, because they are the only one doing photosynthesis. In the very beginning, of berry development, the berries are contributing also because they are doing the photosynthesis, but here they don't do the photosynthesis anymore. So the leaves are producing photosynthetates for the whole plant, but the way that those photosynthetates will be directed can vary. Actually, it vary depending of the location of the leaf, for example. So we usually well, the global advice is that the leaf that is close to the fruit will be the primary provider for photosynthetates for this cluster, for example. But what are doing the other leaves that are on the canopy? Where do they send their photosynthetates? So when you prune the vine, you might redirect those that circuit of phloem, the direction in which things are going. Are the photosynthetates going to the tips to make new shoots, or is it going to go to the roots for sugar accumulation in the roots, for example? So all this highway of photosynthetate well, transit actually in the plant, translocation, this is a process which we call translocation, may be affected by the way that in which you will prune your vine ultimately. But this is something that is not very much studied yet. There's still literature on this, but it's not well known yet. So, but this is just what I wanted to bring to your attention, that this is happening, you know.
Perfect. Yeah, I think you also answered to the second part of the questions, which was, does the berries also perform photosynthesis, not just the leaves? Mm-hmm. You answered
they do contribute in this very first part here, you know.
Yes, thank you. And the other question is, were there any consideration made around the quality of the light and seasonality, for example, UV wavelengths, etc.?
Yeah, so why we decided to pick polycarbonates, if I address the UV light question, of course, we understand that covering the grapes will cut down few UVs, but polycarbonate is pretty good at that. Actually, it's leaving most of the UV in, actually, in the to go through the polycarbonate. This is why we actually chose that material, which is pretty expensive actually in comparison to other plastic that you can use for greenhouses. And we did measure light. So we're not, we don't have the data yet. We're not done with those, all the calculations that are going with it, but we sure measure light, and this is something that we consider. So
perfect.
I don't know if that answers the question.
And the following question, did you notice more vegetative growth in the old treatment?
This I don't have the answer. We do have the, I should ask my student, because my students were in the field all the time. So yeah, so they will be able to answer that question better than me. But I don't remember. Well, we've been controlling the pruning and the crop load itself, and if I remember well, there's no difference in the crop load, but I still I want to see those data first before answering to you. So
perfect.
Sorry.
And the following question, temperature is not affecting the compounds, maybe because there is no water stress during the ripening? That was the slides with the aromatic compound.
Yeah, just a minute, I'll get back to that slide. So the question is the, because there's no water stress, there shouldn't be, they shouldn't have an impact, or is that the question? Or
yes, maybe the reason of not having differences in the aromatic compounds could be the not having water stress during ripening.
Well, we do have differences. That's the thing. The point is that we have, I will have expected to have more of, well, that most aroma compound will be more concentrated in the all treatment, given the fact that this was the most photosynthetically active grapevine, and they had the highest temperature as well. But in contrast, we had higher concentration of compounds in the control, and this is really about abiotic stress for sure. So in general, in plants, when plants are submitted to a stress, could be UV or could be water stress, as the question. In general, a water stress will increase terpenes. It's very well known, and UV stress will increase phenolic compounds, for example. So I can understand that. It sounds logical that some compounds were in a higher concentration in the control due to that. But since we increase the temperature during the berry development time, but you know, I'm not sure I can even say that, because we don't have all the data yet about the pre and post and see if those were affected in the similar way as the control. So I'm not sure I can give an answer right now, but we still need to make our head about those results. But I think that since we had more of those benzyl alcohol and phenyl alcohol in the all treatments, it could be related to the fact that we had some sort of an early development in the plant that were under the greenhouse. And maybe, during the early development, this is at this time that hydroxycinnamates get produced in the berry, that gets accumulated in the berries. And those hydroxycinnamates are precursors for the volatile phenols here. So it could be related, but we're not sure yet. We still need to address those questions with stronger data.
Perfect. Thank you, Karine. You are getting a lot of questions. If you have some more time, I would like to...
I can stay a little bit longer, no problem.
Perfect, thank you.
So it's very nice to have questions. It's always fun when people are interested.
Yes, we have a lot of questions.
If I don't have time to address all the questions, they can write me an email, no problem.
Yeah, sure. Okay, we can answer a couple of more. So the question is, how big a difference do we have in terms of light between all and control?
I don't have the data now, so I cannot answer, sorry.
Perfect. And the next one, do you think the greenhouses influence precipitation or even humidity?
Yeah, we've been doing, we were actually afraid of getting a high humidity in there, and that we would have actually disease problems. And actually we did not get those problems. And it could be because, there's so much wind in Nova Scotia that just the few openings that we had on the sides of the greenhouse were sufficient to have a airflow that just dried out a little bit the plant, well, at least enough to so we don't get mildew or other leaf pathogens. So yeah.
Perfect.
Not sure if I answered the question here, but so for the light, we don't have the data. Sorry, I think I lose a part of the question.
Perfect. And another question, how many growing degree days did each intervention have?
Did each what?
Intervention.
Treatments, you mean?
Yes.
Yeah, I have that on that slide, but this is only the total accumulated growing degree days. So it was about like 1625 here, growing degree days, and it's super high, by the way, because we had an amazingly warm season this year. So so usually we will get much lower growing degree days. Like the year before, this was 2020, and in 2019. When we did that experiment again, but some things turns bad, so we cannot use the results. Yeah, so there was like maybe 1100 growing degree days or 1200 growing degree days during the whole season. But this year, you look at the control here, like almost 15,000 growing degree days. So that's super high. Does that answer the question?
Yes, perfect. And did the polycarbonate affect carbon dioxide levels?
I don't know. That's a good question. You mean in terms of airflow?
Yeah, it didn't really
Could be
specify in there.
Yeah, yeah. I would guess that this is about the airflow here. Yeah, we haven't monitored the CO2, so but this is a very good question, and I will think about it, because since we measure a higher rate of photosynthesis, that's a very good question to address. So thank you.
Perfect. Okay. Let me think. I will just try to choose the ones that are related to your presentation, because we did get a lot of questions. So about the translocation discussion, one of the attendees would like you to comment on nutrient applications to the leaves and timing, if you could.
Well, nutrition in grapevine is typically pretty low, except for, well, especially in hybrid, actually. So I did a project in the past about nutrients, mostly nitrogen and potassium. So in hybrid, typically nitrogen fertilization is supposed to be, well, should stay low, actually, except for some French hybrids that tends to accumulate less nitrogen. With regard to translocation, I'm not sure I can comment on that, but and the timing as well. But what I can tell you regarding the nitrogen, well, any fertilization, is that be careful with the fertilization and the nitrogen fertilization for hybrids, because I saw a lot of very high YAN level in hybrids, and those can well, I saw level as high as 600 milligrams per liter of juice, and this is super high, and it can lead to the formation of certain compounds that are not desirable in the wine, such as ethyl carbamate, for example. And this is a compound that contributes to headaches in the vine, and this is actually a carcinogenic compound that is this is a substance that's controlled by Health Canada. So you don't want to push too hard on the nitrogen, for sure. But you know, hybrids are just usually have a high vigor, so they usually don't need too much nitrogen in general. I'm sure that Francisco can can speak on that better than me, actually.
Perfect. So I will ask you the next 4 questions to finish with, and I apologize, everyone. And as Karine said, if there is any of your questions wasn't answered, you can always reach out to her.
Yeah, send me an email, no problem, or give me a call.
Yeah, perfect. But we can answer maybe the following 4 questions, because they are quite good. So one of them is, do you think flower initiation could be effect for the next year, positively or not, and will you follow that?
If the flower initiation can be affected by the treatment?
Can be affected for the next year.
Yeah, that's a good question, because we haven't considered the year round. I know that that Jim Woodward is working on that topic at Brock University, not to name him, but we haven't considered that aspect. But in general, if you think that the grapevine has was able to carry more photosynthesis and actually gets accumulate some of the value associated to a higher photosynthesis in terms of resources that are stored in the roots, for example, or in the shoots. I would say that it could affect, but this is something we could actually monitor this year. we'll see how well it goes in the vineyard if we go back this spring. But that's a very good question. Thank you.
Perfect. And actually, that was one question. Is a general question, but I think it's important to also highlight that. What was the thought process for selecting L'Acadie versus other cepage popular in Nova Scotia?
Well, this is one of the main varieties that is grown in Nova Scotia. Initially, we wanted to do this experiment on Riesling, but this is unfortunately a variety that is, well, this is a variety that has more value, more commercial value. So we basically take what the growers are ready to give us to play with. So there's a practical aspect to this. But L'Acadie is a very good variety to work with, since it's more or less the emblematic variety here in Nova Scotia. So it's very popular, and it's getting planted. Even in Quebec also, there's a lot of L'Acadie. So I think it was a good choice to start such experiment. We'll see if we can achieve more of this type of experiment in the future in other varieties. The choice is hard, because there's a lot of varieties that are grown here.
That's true. Perfect. And a practical question we have here. If the filter used is so expensive, how would it be used commercially? Do you have any insight on that? The filter used is so expensive. Is there any option for use it commercially?
The filter?
Yes.
It means the polycarbonate.
Oh, okay. Sorry. Okay, no I didn't catch up. No, the goal of this experiment is not to use that commercially, It's really to understand how the grapevine is processing its photosynthetates, or it's doing its biochemistry, what happens to the biochemistry of grapevine when it's submitted to given conditions. So it's not about the process that will be used commercially at all. It will be just a pain, but although I know that there are people growing grapevine under tunnels, for example, even in Quebec, I saw that, and I know that it's done a lot also in Northern Europe, where they use tunnels or even greenhouses to grow grapevine for winemaking. Can you imagine this? So that's pretty interesting. But in our case, it was not an experiment that was aimed at developing a commercial process. It's really to understand better the relationship between grapevine and the climate, and then we can extrapolate that knowledge to terroir. And see in terms of the growing degree days accumulation, days without frost, and what are the patterns of temperature, typical temperature patterns throughout Nova Scotia, for example, to then have a better fit between the varieties and the site. So and I haven't spoke about that part of our work, but we're working a lot about on this relationship actually between the terroir, the climate, and the ripening in a lot of varieties. So we have a lot of results coming up about vinifera and hybrids in Nova Scotia and in Quebec that we've been sampling. So my students are working super, super hard, because every bound aroma sample is like a 3-day extraction until they can actually inject the sample in the GCMS for the aroma characterization. So it's a very long process, but it's going to come at a time. So and it will be very exciting to present you those results.
Perfect. Thank you very much, Karine. We literally bombarded you with with questions. Thanks, everyone.
I'm very glad. I'm super happy, because it means that this is interesting. So
yes, you do have more questions, and I would encourage people to contact you for further questions.
Yeah, don't be shy. You can contact me anytime, no problem.
Perfect. Thank you very much.
I'm very used to have growers calling me for a question, sending me an email for any question, no problem.
Thank you very much, Karine. I will stop sharing your screen.
Yeah.
Share this just to finish. Thank you very much again for the presentation. As Nestle said, we provide you a lot of questions. People who didn't have their question right now, my apologies, but we are already past time. So thank you very much. Just to before closing this session, join us again for the next session, which will be in January 28th of 2021 with Dr. Belinda Kemp from Cody, Ontario, and her master student candidate, Hannah Charnock. They will talk about the influence of the type of sugar used and the metal content on the flavor of sparkling wines. Okay, so keep in mind that date. We will be sending you an email with a survey. Please, we encourage you to answer the survey so we can be improving these sessions in the future. And also we'll be providing you the link to your register. Okay, so again, thank you.